Hi, my name is Victor Alvarado, and this is my research project. It's on the comparative analysis of Zamia leaf anatomy and vulnerability to herbivory damage. Now, this is going to be a rough overview of the topics that I'm going to talk about today. Now, on to the first thing, the Zamias. Zamias are a subgenus of the Cycadales order. They are a group of gymnosperm organisms, which means that their seeds grow exposed to the atmosphere. They're not protected by a fruit or an ovary. These plants have existed for over 300 million years since the Permian period. In the Zamia genera, approximately 86 species to date have been identified and studied to a certain degree. This information came from the World List of Cycads website. Now, like any other plant, these cycads interact with their environment through pollination, resource competition, nutrient cycling, herbivory, and etc. Some of these interactions are influenced by unique characteristics of the plants, which can be the size, the growth rate, and the leaf patterns of these plants, and the anatomy of the plants. Now, on to why I chose some of the species that are for this study. Now, the four species I have chosen, as you can see, are the Zamia integrifolia, the Zamia vasquezi, the Zamia decumbens, and the Zamia furfuracea. I included where they are native to and their current conservation status. Now, these Damias cycads overall grow very slowly and unpredictably, so they may be very vulnerable to endangerment if they are all decreasing in population numbers. So conservation needs to be something that is very enforced. This is the Humus atala butterfly, also known as the Atala hairstreak butterfly. It is the most prevalent herbivore, if not the only ones, of the Zamicycads. The Zamia integrifolia species is specifically the host plant of this butterfly. The adult female talas will seek out new leaves that are growing on these cycads where they will lay their eggs. Now, some chemical pest measures were considered as the beneficial predators, introducing other animals to eat these butterflies. The reason this didn't work out is because the cycads contain a toxin called cycacin, which is found mostly in the cones of the plants, but they are found throughout the body, the leaves, and everything. And these caterpillars have evolved to be able to eat this toxin, contain it in their body throughout their lifetime. Now, the overpopulation of these butterflies. Normally, these cycads have a set flush period throughout the year, which means that only throughout a couple months of the year will they start growing new leaves, and that is typically when is expected for the butterfly populations to also increase. But the issue where that lies here is in Montgomery Botanical Center, several Zami species are grown, protected, and studied. Now, the collection of all of these Zamia species in one collected area has led to a boom in the tala populations, typically because cycads in general from all around the world have different flushing periods, which means they grow leaves during different times of the year. So the atalas always have a food source available. The research question for this project was, how does the epidermal anatomy of these Caribbean Zamia species affect herbivory by the lepidopteran larvae? The objectives of this project were to perform a comprehensive comparison of the four Caribbean Zami species and to identify possible trends or similarities in the epidermal layer of the leaflets using SEM techniques. Now, for the materials and methods, in total, about 24 samples were collected from Montgomery Botanical Center. The 8 initial samples were collected on September 15, where they were stored in a herbarium oven at a temperature of 43 degrees Celsius for 5 days, after which they were stored in silica gel bags and transported to the analytical electron microscopy lab in FIU. As you can see here, this is one of the machines used in the initial sample preparation steps. A quarter-inch cuttings of the leaflets were made. They were stubbed with double-sided tape on these stubs right here, and they were inserted into the sputter coater in order to make the samples conductive. For the second round of sample preparation, the rest of the 16 samples were collected and similarly stored in silica gel and taken here to the lab in FIU. Now, like before, a quarter inch to about 2/3 of an inch cuttings were made from these leaves. They were fixed in 99.9% methanol for 10 minutes to fix the epidermal cell structure, which were followed by two 30-minute dehydration sequences in the anhydrous ethanol. This is a critical point drying machine that was used still in the second part of the sample preparation. The samples, after being fixed in methanol and ethanol, they were put into the critical point drying machine, which the methanol was used as a transitional fluid and liquid carbon dioxide was used as a bleeder. Now, these are the young adaxial leaflets. As you can see here, I have labeled some of the anatomical features with the arrows and the letters next to them. But as you can see, one similarity that they all contain is they have trichomes. These are non-glandular unicellular trichomes that they can serve many purposes, which I will get to more into in the discussion portion. As you can see here, they have different anatomical features. These are the pavement cells that all of the species seem to have except for the Furfuracea, which is basically you cannot see the epidermal surface of these leaves because their trichome density is just so intense. These are the adult adaxial sides of the leaf. And as you can see here, this is where they lose many of their distinctive features. They all kind of look similar except for Vasquezi, which they have undulate pattern of their pavement cell structures. As you can see, the most drastic change, however, from young to adult leaflets is the Furfuracea, as they have seemed to lose almost all of their trichomes, and you can finally see the actual surface of the leaflet. These are the young abaxial side of the leaflets. As you can see, the Furfuracea looks the same, and for the young antigrafoliate leaflet, you can see the pattern of stomatas following the one row going down. As you can see here, I'm not sure if you can see very well because it is zoomed out. This is the parallel venations going down, and then these are the stomatal bands going down. And one thing that I also did not discuss is that for the cycads and Zamia species specifically, no species has to date has been studied to have stomatas on both sides of the leaflet, mainly on the abaxial side, but not on the adaxial side. And as you can see here, this is where the integrafolia stands out. As you can see, there are subsidiary cells, guard cells, and ultimately stomatas found on the adaxial side hidden behind the trichomes. Now, these are the adult abaxial surfaces of the leaflets, and this is where they all look very similar, but this is where you can see the prominent like distinctions, although Enterofolia and decumbens, as well as the Furfuracea, they have a similar patterns, the pattern, the parallel venation, and the many rows and columns of stomatal bands going down. But the Zamia vasquezis, as you saw in the young leaflets as well, they had a very prominent pavement cell structure. They share this feature as well in the abaxial side. As you can see, it's kind of like a puzzle shaped manner. Now, what may some of these features describe about the plant specifically? For the stomatas found on both faces of the young intercofoliate leaf, I found studies linking plants that have increased nitrogen levels because of the cyanobacteria that live in their roots. And some of these increased nitrogen levels have been linked to increased rates of photosynthetic activity, which may explain why the plants may need to or may have adapted to grow the maximum amount of stomatas in their young leaflets. But it does not explain why the adult adaxial and tegrifolia face lost all stomatal presence throughout its maturation. The Zamia cousins, the Zamia decumbens and Vasquezi, their unique pavement cell structures may not indicate a connection to herbivory damage, but they may be adaptations to environmental mechanical stresses such as dry soils, intense rains, and intense exposure to wind. And the high trichome density observed in Zamia furfuracea leaflets could mainly be the only really defensive adaptation seen amongst all the imaging that I did that may be against pests, specifically the caterpillars, as this trichome density, once thick enough in the young leaflets, has the possibility to affect egg laying behavior or feasibility. Of caterpillars and the movement through the young leaflets may also be more difficult as they would highly increase the surface area, so it may not be easy to chew through the leaflets. Now, the importance of these anatomical features as pavement cells, as we saw in most of the imagings, they are irregularly shaped parenchymal cell barriers between the atmosphere and the specialized cells below. The guard cells control the opening and closing of the stomatal pores to facilitate gas exchange and transpiration. The trichomes, as I talked about, they are an additional protective layer against abiotic and biotic hazards. The subsidiary cells help increase the function and protection of guard cells, as they are also known as accessory cells of the plant. The stomata, they help with the intake of carbon dioxide and they also release oxygen, which is the gas exchange portion, and they help control water vapor flow. Now, how may some of these anatomical features connect to herbivory interactions? I have seen that stomatal quantity and the act of them being closed or open can help affect the leaf temperature of the plant of the leaf, as well as the water and nutrient content, which can affect how viable of a food source it is for pests. Trichomes also have the ability to facilitate chemical defenses in response to being eaten. Some future directions I would like to take this project is doing a carotenoid analysis and identification of ketocarotenoids in relation to these Humus butterflies being linked to having adapted to grow red wavelength vision, as they can see red when most other butterfly species are only able to see UV, blue, and green. I also want to do different environmental conditions effects on the leaf anatomy of these Zamia species, which means that exposing them to different concentrations of salt levels, different concentrations of light intensity, and different concentrations of nutrients. Now, for the semiochemical analysis is to do studies on the sex pheromones of these specifically on the female Lepidoptera natalis in combination with the host plant volatiles of the plant. Help disrupt the mating process, as the sex pheromones could then be used to confuse the male Lepidoptera and butterflies. These are the acknowledgements, the people that helped me throughout the project. These are the references that I used. Thank you.